Welcome back to the deep dive. Today, we are taking a necessary breath literally. We're not just looking at the simple action of breathing in and out.
No, not at all. We are doing a really extensive deep dive into the incredibly complex system that keeps every single one of our cells running. Human respiration.
Exactly. And it's more than just, the plumbing. It's chemistry, it's physics, it's control systems, all working together.
Absolutely. Our mission today is pretty clear. We are diving deep into the NCERT material. We want to understand the precise mechanics, the subtle chemistry, and that really tight neural regulation that governs how we exchange oxygen and carbon dioxide.
So the goal is that you, the listener, should walk away from this with a conceptually complete grasp of this whole vital process.
That's the aim. And that word integrated you used is crucial.
Yes. Before we jump into the lungs and the blood vessels, let's talk about this idea the NCERT brings up, systems biology.
It's a really important perspective. Traditionally, science, especially biology, often used a reductionist approach. You break things down.
Right. Look at one molecule, one gene, one cell type in isolation.
Exactly. And look, that approach has given us a huge amount of knowledge, especially in molecular biology, but it can miss the bigger picture.
How things actually work together in a living organism.
Precisely. The NCERT points out that biological systems are often much more than just the sum of their parts. The real magic, the things we observe as life, these are often emergent properties.
Emergent properties, meaning they arise from the interactions.
That's it. When molecules bump into each other in the right way, when cells cooperate to form tissues, when organs coordinate their functions, the resulting system can do things the individual bits just can't.
And the respiratory system is a prime example.
Oh, absolutely. Think about it. You've got pressure changes, complex chemical transport, precise timing signals from the nervous system, all happening across different organs and tissues. It's the perfect illustration of this kind of complex integration.
That really sets the stage nicely. And speaking of how different physiological studies connect, the NCERT mentions Alfonso Corti, an Italian anatomist.
Yes, born 1822. He's famous for his work on hearing, actually. The mammalian auditory system.
The organ of Corti.
That's the one. He described this incredibly intricate structure within the cochlea back in 1851. It's the part that converts sound vibrations into nerve signals.
Okay, but why mention him in a deep dive on breathing?
Well, think about his work. He was intensely focused on how a highly specialized, very delicate membrane structure performed a specific function converting mechanical energy to neural signals, understanding that kind of specialized membrane function.
I see. It kind of paves the way intellectually for understanding other specialized membranes.
Exactly, like the incredibly thin, multi-layered diffusion membrane in our alveoli, which we'll get to. It just highlights how insights from studying one complex biological system can really unlock understanding in another. It's all interconnected.
Okay, let's get down to basics then. The fundamental why. Why do we actually need to breathe oxygen constantly?
Well, the NCERT lays it out clearly. Organisms use oxygen, O or O, to indirectly break down simple fuel molecules, things like glucose, amino acids, fatty acids.
The catabolic process, right? Breaking things down.
Yes, to release the energy stored in those molecules, energy that's needed for, everything life does.
But that breakdown isn't perfectly clean, is it? There's waste.
There is. It inevitably produces a harmful byproduct. Carbon dioxide, QOS. So respiration at its core is this continuous, absolutely vital exchange. We need a constant supply of ORO coming into the cells.
And a constant removal of the co-ORO going out.
Precisely. Life depends on it moment to moment.
What's really fascinating, though, is how differently life forms have solved this exchange problem. The NCERT walks us through this evolutionary journey.
It's quite a showcase of adaptation. You start with the simplest forms, lower invertebrates like sponges, coelenterate, flatworms. They don't have complex organs for this.
So they just absorb it.
Pretty much. Simple diffusion across their entire body surface. If you're small and simple enough and maybe living in water, that works.
Then things get a bit more specialized, like earthworms.
Right. Earthworms use cutaneous respiration. They breathe through their moist cuticle, their skin, essentially. Amphibians, like frogs, also do this. They use their moist skin to supplement what they get through their lungs.
Okay, what about insects? They have those tiny holes, right?
Spiracles, yes. They lead to a tracheal system. It's this amazing network of tubes that basically pipes atmospheric air directly to the tissues deep inside the body. Very efficient for their size and lifestyle.
And in water. You can't just diffuse air easily.
No. So you see the evolution of gills, that's branchial respiration. These are highly vascularized structures full of blood vessels designed to maximize gas exchange with water, found in aquatic arthropods, mollusks.
And, of course, fish.
And then for life on land, especially for larger, more active animals like reptiles, birds, and us mammals, we developed pulmonary respiration using lungs.
These large internal vascularized air sacs, which brings us neatly to the human system. Let's trace that pathway, as the NCERT details it.
Okay. Air enters through the pair of external nostrils. Then it goes into the nasal chamber, through the nasal passage.
And into the pharynx.
Right. And the pharynx is interesting because it's a crossroads.
Yeah, it's a common passage for both food and air, which sounds potentially hazardous.
It requires very precise control. From the pharynx, the air pathway continues through the larynx region and into the trachea.
The larynx, that's the voice box.
That's it, the cartilage and a sound box. But crucially, guarding the entrance to the larynx, the glottis, is the epiglottis.
That thin, elastic cartilaginous flap?
Yes. Its job is vital. During swallowing, it flips down like a lid, covers the glottis, and makes sure food goes down the esophagus, not into the airway, the trachea.
A critical safety feature. So once air is safely past the larynx, it's into the trachea.
The trachea, yes. A straight tube that runs down into the middle of the chest, the mid-thoracic cavity. The NCERT is very specific. It divides into the right and left primary bronchi at the level of the fifth thoracic vertebra.
Okay, T5. And this is where the branching really begins, the bronchial tree.
It is. The primary bronchi divide into secondary bronchi, then tertiary bronchi, and these keep dividing into smaller and smaller tubes, ending in the very fine terminal bronchioles.
And these tubes need to stay open, right? Even when pressure changes.
Absolutely. That's why the trachea, the bronchi, and even the initial parts of the bronchioles are supported by these incomplete rings of cartilage. They act like scaffolding, keeping the airways patent, preventing collapse.
So this whole network, this conducting system, ultimately delivers the air where?
To the alveoli. These are the business end of the system. They're described as very thin, irregular-walled, highly vascularized, sort of bag-like structures, millions of them.
And this whole structure, bronchi, bronchioles, alveoli, makes up the lungs.
The two lungs, yes. And they're housed within the thoracic cavity, protected by a double-layered membrane called the pleura.
With pleural fluid in between.
Yes, a thin layer of pleural fluid. This is essential. It acts as a lubricant. As the lungs inflate and deflate 12, 16 times a minute, this fluid drastically reduces friction between the lung surface and the chest wall.
Makes sense. And there's an outer and inner pleural layer.
There is. The outer pleural membrane is in close contact with the thoracic lining, the inside of the rib cage. The inner pleural membrane adheres directly to the surface of the lungs themselves.
Okay, so the NCERT then breaks this down functionally. Two main parts based on what they do.
Correct. First, you have the conducting part. This includes everything from the external nostrils right down to the terminal bronchioles.
And its job is basically transport and preparation?
Exactly. Transporting the atmospheric air to the alveoli, but also filtering it, clearing out foreign particles, humidifying it, and bringing it up to body temperature, getting it ready for exchange.
And the second part?
That's the exchange part. This consists of the alveoli and their associated ducts. This is where the actual action happens, the diffusion of O and Koyoro between the blood in the capillaries and the air in the alveoli.
And this entire sits within the thoracic chamber. You mentioned its structure earlier.
The anatomy of this chamber is key to the mechanics of breathing. You've got the vertebral column at the back, dorsally, the sternum or breastbone at the front, ventrally, the ribs wrapping around the sides, laterally, and the large dome-shaped diaphragm muscle forming the floor, inferiorly.
And it's an airtight chamber. That's crucial, isn't it?
Absolutely critical, because the lungs themselves don't have significant muscle to change their own volume directly. They're passive in that sense.
So any change in the volume of the lungs has to follow a change in the volume of the thoracic chamber.
Precisely. The NCERT emphasizes this. We cannot directly alter pulmonary volume, but by changing the volume of the surrounding airtight thoracic chamber using muscles like the diaphragm and intercostals, we force the lungs to expand or contract along with it. That's what creates the pressure changes that drive air movement.
Okay, we've got the structure, the house where breathing happens. Now let's look at the mechanics, how we actually ventilate that house. The NCERT outlines 5 main stages in the whole process of respiration, right?
Yes, it's good to keep the overall sequence in mind. First is what we commonly call breathing or pulmonary ventilation, getting air in and out of the lungs.
Step 1.
Step 2 is diffusion of gases, O and Co euro, across that thin alveolar membrane we just discussed. Step 3 is the transport of these gases by the blood to and from the tissues.
Okay.
Step 4 is diffusion again, but this time between the blood and the actual body tissues. And finally, step 5 is the utilization of O euro by the cells for metabolism, cellular respiration, which of course produces the CRO.
Got it. So let's focus now on step 1, the mechanism of breathing. This involves 2 phases, inspiration and expiration.
Taking air in and letting air out. And the fundamental principle, as we touched upon, is creating pressure gradients. Air flows from higher pressure to lower pressure.
Just like any fluid. So for inspiration, to get air in, the pressure inside the lungs, the intrapulmonary pressure, has to be
lower than the atmospheric pressure outside. You need to create a relative negative pressure, a slight vacuum.
And how do we do that? Which muscles are the key players?
It's primarily a 2-part muscular effort for normal, quiet inspiration. First, the diaphragm. This large, dome-shaped muscle at the base of the thorax contracts.
And when it contracts, it flattens and moves downward.
Exactly. That movement significantly increases the volume of the thoracic cavity in the vertical or antero-posterior axis. Think top-to-bottom increase.
Okay, one dimension covered. What's the second part?
The external intercostal muscles. These are muscles situated between the ribs. When they contract, they pull the ribs and the sternum upwards and outwards.
Ah, so that increases the volume side to side and front to back, the dorsoventral axis.
Precisely. So the contraction of the diaphragm and the external intercostals causes an overall increase in the volume of the thoracic chamber.
And because the lungs are stuck to the inside of this chamber via the pleura,
the lungs are stretched, their volume increases too. An increase in volume leads to a decrease in pressure inside the lungs, dropping it just below atmospheric pressure. Air then rushes in down the pressure gradient. That's inspiration.
Clever. So what about expiration, getting air out?
For normal, quiet expiration, it's typically a passive process. It happens when the intrapulmonary pressure becomes slightly higher than atmospheric pressure.
Passive. So no muscle contraction needed.
Generally not for quiet breathing. The diaphragm simply relaxes, returning to its dome shape. The external intercostal muscles relax, letting the ribs and sternum fall back to their resting positions.
Which reverses the volume changes.
Yes. The thoracic volume decreases, which in turn decreases the pulmonary volume. This slight compression increases the pressure inside the lungs to just above atmospheric, and air flows out.
But the NCERT mentions we can increase the strength of breathing using abdominal muscles. When would we do that?
That's for forced breathing. Think about activities like vigorous exercise, shouting, coughing, or even playing a wind instrument. You need to move air more forcefully or quickly.
So for forced expiration.
Yes. For forced expiration, you actively contract abdominal muscles. This pushes the abdominal organs upwards against the relaxing diaphragm, further reducing thoracic volume much more rapidly and completely than passive relaxation alone. You can also use the internal intercostal muscles to pull the rib cage down and in more forcefully.
And for forced inspiration.
You can use accessory muscles in the neck and chest to lift the ribs even higher, maximizing the thoracic expansion beyond what the diaphragm and external intercostals do alone.
Okay, that makes sense. And the NCERT gives a baseline rate a healthy person breathes about 12 to 16 times per minute.
That's the typical resting rate. And clinicians can measure how efficiently this whole mechanical pump is working using a device called a spirometer.
Right, the spirometer measures the volumes of air moving in and out. This gives us those important respiratory volumes and capacities.
Exactly. These measurements are really valuable for assessing lung function and diagnosing potential problems. Let's go through them, as defined in the NCERT.
Let's start with the most basic, tidal volume, or TV.
Tidal volume is the amount of air you breathe in or out during a normal, quiet breath. It's surprisingly small, only about 500 milliliters. 500 milliliters?
500 ml per breath, but over a minute.
Well, if you breathe 12 times a minute, that's 12 times 500, which is 6,000 milliliters, or 6 liters per minute. If you breathe 16 times, it's 8,000 milliliters, or 8 liters. So the minute volume is about 6,000 to 8,000 ml.
Okay. Next up is the inspiratory reserve volume, IRV. The reserve.
This is the extra amount of air you can forcibly inhale after you've already taken a normal tidal breath in. It's quite substantial, averaging around 2,500 to 3,000 milliliters. 2,500, 3,000 milliliters. Think of it as your deep gasp capacity.
And the opposite must be the expiratory reserve volume, ERV.
Correct. That's the additional volume of air you can forcibly exhale after normal tidal breath out. This averages about 1,000 to 1,100 milliliters, 1,000, 1,100 milliliters.
So TV is normal breathing, IRV is extra in, ERV is extra out. What about what's left?
Yes. Even after you've forced out as much air as you possibly can after exhaling your ERV, there's still some air left in the lungs. This is absolutely crucial. It's called the residual volume, or RV.
And why is it crucial? Why not empty the lungs completely?
Because if the lungs emptied completely, the tiny alveoli could collapse shut, like wet plastic bags sticking together. Reinflating them would take immense effort and could cause damage. The RV, which is about 1,100 to 1,200 milliliters, 1,100 to 1,200 milliliter, acts as a buffer, keeping the alveoli partially inflated and allowing continuous gas exchange even between breaths.
Okay, that makes perfect sense. So those are the 4 basic volumes. Now, how do they combine into the capacities used clinically?
Right. The capacities are essentially sums of 2 or more volumes. First, inspiratory capacity, IC. This is the total amount of air you can inhale starting from the end of a normal exhalation. So it's your normal tidal volume plus your inspiratory reserve volume, TV plus IRV.
And its counterpart.
Expiratory capacity, EC. That's the total amount you can exhale starting from the end of a normal inhalation. Logically, that's tidal volume plus expiratory reserve volume, TV plus ERV.
Okay, IC and EC. What's next?
Functional residual capacity, FRC. This is a really important one clinically. It's the volume of air remaining in the lungs after you've finished a normal, quiet exhalation. So it's the air normally left in the lungs between breaths, your expiratory reserve volume plus your residual volume, ERV plus RV. It reflects the resting state of the lungs.
FRC, ERV plus RV. Got it. 2 more major ones.
Yes. Vital capacity, VC. This represents the maximum amount of air you can possibly move in or out of your lungs in one breath. It's defined as the maximum volume you can exhale after taking the deepest possible inhalation.
So that would be everything except the residual volume.
Exactly. VC is your expiratory reserve volume plus your tidal volume plus your inspiratory reserve volume, ERV plus TV plus IRV. It's a key measure of the usable volume of your lungs.
And finally, the big one, total lung capacity, TLC.
As the name suggests, this is the absolute total volume of air the lungs can hold after the biggest possible inhalation. It includes everything. The residual volume plus the expiratory reserve volume plus the tidal volume plus the inspiratory reserve volume, RV plus ERV plus TV plus IRV. Or, more simply, it's the vital capacity plus the residual volume, VC plus RV.
Wow. So understanding these volumes and capacities gives doctors a detailed picture of lung health.
A very detailed picture, yes. Changes in these values can indicate various respiratory diseases or conditions affecting lung mechanics.
All right, we've ventilated the lungs using mechanics. Now, the crucial step, getting the oxygen out of the air and into the blood, and the carbon dioxide out of the blood and into the air. Where does this happen?
The primary site, overwhelmingly, is the vast surface area provided by the alveoli. There's also some exchange that happens between the blood and the body tissues, of course, but the lungs are where atmospheric gas exchange occurs.
The mechanism is fundamentally simple.
It's surprisingly simple, yes. It's simple diffusion. Gases just move down their concentration, or, more accurately, their pressure gradient.
But it's not just the gradient, right? The NCERT mentions other factors.
Correct. While the pressure gradient is the main driver, 2 other factors are critical. The solubility of the gases in the fluid lining the alveoli and in the blood plasma, and the thickness of the membrane they have to cross.
Okay, let's unpack the pressure gradient first. We need to talk about partial pressure.
Yes. Air isn't just oxygen. It's a mixture of gases, mostly nitrogen, then oxygen, a tiny bit of carbon dioxide, and other trace gases. The partial pressure of a gas, like oxygen, written as PO, is the pressure that specific gas contributes to the total pressure of the mixture.
So it's like oxygen's individual share of the total atmospheric pressure.
Exactly. And gases always diffuse from an area where their partial pressure is higher to an area where their partial pressure is lower.
Let's use the numbers from the NCERT's table 14.1 to see this in action. What's the PO euro in the air we breathe?
In the atmospheric air, the POO is about 159 millimeters of mercury. 159 millimeters of mercury.
But by the time that air reaches the alveoli, it mixes with the residual air, it gets humidified.
Right. So the PO in the alveolar air drops to about 104 millimeters of mercury. 104 millimeters of mercury. Now compare that to the blood arriving at the lungs from the body.
That's deoxygenated blood, right? It's dropped off its oxygen to the tissues.
Precisely. That blood has a very low PO, only about 40 millimeters of mercury, 40 millimeters Hg.
Okay, so we have 104 millimeters of mercury, 104 minus 40. This steep gradient powerfully drives oxygen from the alveolar air across the membrane and into the blood plasma and red blood cells.
Until the blood gets oxygenated. What's the PO then, leaving the lungs?
The blood leaving the lungs, now oxygenated, typically has a PO of about 95 millimeters of mercury. 95 millimeters of mercury. Not quite 104 due to some minor mixing, but very high.
And this oxygen-rich blood goes to the tissues. What's the PO there? Well, the tissues are constantly using oxygen for metabolism, so the PO within the tissue cells is low, around 40 millimeters of mercury. 40 millimeters Hg.
Ah, so now the gradient reverses direction, in a sense.
Yeah.
High PO in the blood, 95, and low PO in the tissues, 40.
Exactly. So oxygen readily diffuses out of the blood capillaries and into the tissue cells where it's needed.
Okay, that makes sense for oxygen. Now, what about carbon dioxide, the waste product? Let's trace its journey. Where is its partial pressure highest?
In the tissues where it's being produced by metabolism, the PCO in the tissues is around 45 millimeters of mercury, 45 millimeters of mercury, 45 millimeter Hg.
And the oxygenated blood arriving at the tissues.
That blood has a lower PCO, about 40 millimeters of mercury, 40 millimeter Hg. So PCO diffuses out of the tissues from 45 into the blood to 40.
Okay, so the blood picks up KO at the tissues. Now this deoxygenated CoA-rich blood travels back to the lungs. What's its PCO?
It arrives at the lungs with that higher PCO of about 45 millimeters of mercury, 45 millimeters Hg.
And what's the PCO in the alveolar air, waiting to be breathed out?
The picouros in the alveoli is about 40 millimeters of mercury, 40 millimeter Hg.
Wait a second, let me get this straight. The PO gradient driving oxygen in at the lungs was huge, 64 millimeter Hg. But the PO euro gradient driving carbon dioxide out is only 5 millimeter Hg, 45 down to 40.
That's correct. The pressure gradient for co-euro exchange at the lungs is much, much smaller than for O-euro.
How can that possibly be efficient enough? If the push is so much weaker, how do we get rid of co-euro effectively?
This is where the second factor comes in solubility. The NCERT highlights this as absolutely crucial.
Solubility, how easily the gas dissolves.
Yes. And carbon dioxide is vastly more soluble in water, and therefore in the plasma and fluid lining the alveoli, than oxygen is. The NCERT states QO solubility is about 20 to 25 times, 25 times higher than that of O.
20 to 25 times higher. Wow.
So even though the pressure difference pushing Kunko is small, only 5 millimeter Hg, because it dissolves and moves across the membrane so much more easily than ORO, the amount of CoO that diffuses per minute is still sufficient to match the body's needs. Its high solubility effectively compensates for the low pressure gradient.
That's incredible. It's like CoOs doesn't need a strong push because the door is wide open for it, whereas ORO needs a harder push because its door is narrower.
That's a pretty good analogy, yes. Solubility makes a massive difference.
Okay, gradient and solubility. What was the third factor?
The thickness of the diffusion membrane, the barrier the gases have to cross between the alveolar air and the capillary blood.
And presumably it needs to be very thin.
Extremely thin. The NCERT emphasizes its total thickness is much less than a millimeter. It's made up of 3 main layers.
3 layers. What are they?
First, the wall of the alveolus itself, which is made of a single layer of very thin, flat cells called squamous epithelium.
Layer 1. Alveolar wall.
Second, the wall of the capillary blood vessel, which is also just a single layer of celled endothelium.
Layer 2. Capillary wall.
And third, sandwiched between these 2 cellular layers, is the basement substance. This is basically a very thin layer of extracellular material, essentially the fused basement membranes of the alveolar epithelium and the capillary endothelium.
So it's basically 2 single-cell layers with a thin bit of connective glue in between.
That's a good way to think of it. Incredibly thin, maximizing the rate of diffusion.
So to sum up this section, we have favorable pressure gradients driving O in and CO out. We have CoER's amazing solubility compensating for its smaller gradient, and we have an incredibly thin diffusion membrane.
Exactly. The NCERT concludes that all these factors in our respiratory system are ideally suited for efficient diffusion of OUR from the alveoli to the tissues, and for SUORs from the tissues back to the alveoli. The physics and the structure are perfectly matched.
Okay, the gases have crossed the membrane at the lungs. Oxygen is in the blood. Carbon dioxide is heading out. Now, how does the blood actually transport these gases around the body? This isn't just simple dissolving, right?
Not for the most part, no. Simple dissolving in plasma plays a very minor role, especially for oxygen. The blood uses highly specialized chemical mechanisms, mainly involving red blood cells and the protein hemoglobin.
Let's start with OGO transport. How is oxygen carried?
The overwhelming majority, about 97%, according to the NCERT, is transported bound to hemoglobin within the red blood cells, RBCs.
97%, so only about 3%. 3% is just dissolved in the plasma.
That's right. Plasma just can't carry enough dissolved oxygen to meet the body's metabolic demands. We absolutely rely on hemoglobin.
And hemoglobin is that red iron-containing pigment in RBCs.
Yes. It binds reversibly with oxygen. That reversibility is key. It needs to pick up OGURO easily in the lungs and release it easily in the tissues. When oxygen is bound, the molecule is called oxyhemoglobin.
How much oxygen can one hemoglobin molecule carry?
Each hemoglobin molecule has 4 iron-containing heme groups, and each heme group can bind 1 molecule of OO. So one hemoglobin molecule can carry a maximum of 4 OO molecules when fully saturated.
What controlled whether hemoglobin binds ARO or releases it?
The primary factor is the partial pressure of oxygen itself, the PO. Where PO is high, like in the lungs, hemoglobin readily binds oxygen. Where PO is low, like in the tissues, hemoglobin releases oxygen.
But the NCERT says other factors are also involved.
Yes, several other factors influence how tightly hemoglobin holds onto oxygen. These include the partial pressure of carbon dioxide, PCO, the concentration of hydrogen ions, which determines acidity or pH, and temperature.
How do these factors affect binding?
Generally, higher pico-euro, higher A concentration, meaning lower pH, more acidic, and higher temperature all decrease hemoglobin's affinity for oxygen, causing it to release ORO more readily.
Which makes perfect sense for the tissues, right? Active tissues produce more EOREs, more acid, like lactic acid during intense exercise, and more heat.
Exactly. These conditions signal to hemoglobin, this tissue is working hard, release the oxygen here. It's a beautifully regulated delivery system.
This relationship is shown graphically, isn't it? The oxygen dissociation curve.
Yes, if you plot the percentage saturation of hemoglobin with oxygen against the partial pressure of oxygen, you get a characteristic S-shaped or sigmoid curve. This curve isn't linear. Its shape is really important.
Why F-shaped?
It reflects cooperative binding. Once the first oxygen molecule binds to hemoglobin, it slightly changes the shape of the molecule, making it easier for the second, third, and fourth oxygen molecules to bind. And conversely, when one ORO leaves, it makes it easier for the others to leave.
And the position of this curve can shift.
Yes. Factors like high CoO, HURO, and high temperature shift the curve to the right. This means that for any given PO, the hemoglobin will be less saturated. It has released more oxygen. This happens in the tissues.
And in the lungs.
In the alveoli, conditions are the opposite. High POROs, low PEOO, lower H concentration, slightly lower temperature. These conditions shift the curve to the left, meaning hemoglobin binds oxygen more tightly and becomes highly saturated, favorable for loading up.
So the curve shifts left in the lungs for pickup and shifts right in the tissues for drop-off.
Precisely. The difference in saturation between the blood leaving the lungs around 95–97% saturated and the blood returning from the tissues around 70–75% saturated at rest tells us how much oxygen was delivered.
And the NCERT quantifies this delivery.
It does. Under normal physiological conditions, every 100 milliliters, 100 milliliter of oxygenated blood delivers approximately 5 milliliters of ORO to the tissues as it passes through the capillaries. This increases significantly during exercise when tissue PO drops lower and the rightward shift is more pronounced.
Okay, that covers oxygen transport, heavily reliant on hemoglobin. What about QO transport? It's more complicated, isn't it?
It is definitely more complex. CoO is transported in the blood in 3 main forms, according to the NCERT.
3 forms. What are they?
First, like oxygen, a small amount is simply dissolved in the plasma, about 7%. 7%.
Okay, 7% dissolved.
Second, about 20–25% binds to hemoglobin, but not to the iron atom where oxygen binds. Co-Euros binds to amino groups on the protein part of hemoglobin, forming a compound called carbaminohemoglobin.
Carbaminohemoglobin. So hemoglobin carries both O and co-Euro, but at different sites.
Yes, which is important. And the binding of co-Euro to hemoglobin is also influenced by partial pressures. It binds more readily when pico-Euro is high and PO is low, like in the tissues, and is released when pico-Euro is low and PO is high, like in the lungs.
That sounds convenient. Low oxygen helps K-Euros bind. High oxygen helps co-Euro release.
It is. This interaction is related to the Haldane effect, which we can touch on. But first, the third and most important form of co-Euro transport.
The biggest chunk.
Yes, the vast majority, about 70%, is transported in the blood plasma as bicarbonate ions.
Bicarbonate. How does QO get converted into bicarbonate?
This involves a crucial chemical reaction, greatly accelerated by an enzyme called carbonic anhydrase. This enzyme is found in very high concentrations inside red blood cells, but only in tiny amounts in the plasma.
Carbonic anhydrase, what reaction does it speed up?
It catalyzes the rapid combination of QO with water, H0, to form carbonic acid, H0 plus H0, H0 for Euro. This carbonic acid is unstable and quickly dissociates into a bicarbonate ion and a hydrogen ion HCO, HCO plus H.
So QOs plus water makes carbonic acid, which splits into bicarbonate and a hydrogen ion, and this happens fast inside the RBCs.
Extremely fast, thanks to carbonic anhydrase. So at the tissues, where PICO is high, QO diffuses into the RBCs. The enzyme instantly converts it to carbonic acid, which then dissociates. The bicarbonate ions, H2O, are then often transported out of the RBC into the plasma in exchange for chloride ions moving in, this is called the chloride shift, to maintain electrical balance. The hydrogen ions mostly get buffered by binding to hemoglobin inside the RBC.
Buffering the He ions is important, right? Otherwise, the blood would become very acidic.
Absolutely critical. Hemoglobin acts as a major buffer in this process. By trapping Key-Euros as bicarbonate in the plasma and buffering the resulting He ions within the RBCs, the blood can carry large amounts of TO from the tissues without a drastic drop in pH.
Okay, so that's how TO is carried from the tissues. What happens when this blood reaches the alveoli?
At the alveoli, the PO in the air is low. This causes all those reactions to reverse. Bicarbonate ions move back into the RBCs, chloride moves out. They combine with the hydrogen ions released from hemoglobin, especially as oxygen binds to it. This reforms carbonic acid.
And then carbonic anhydrase steps in again.
Yes. It rapidly converts the carbonic acid back into Kioro and water. This newly formed Kioro then diffuses out of the RBC, across the capillary wall, across the alveolar wall, and into the alveolar air, driven by the P-thoro gradient, from 45 in blood down to 40 in alveoli.
Wow. It's a reversible chemical shuttle powered by an enzyme and driven by partial pressure gradients.
Exactly. And remember that interaction we mentioned. The Haldane effect describes how the oxygenation state of hemoglobin affects its key RUO carrying capacity. Deoxygenated hemoglobin in the tissues is better at binding both Ki-O, as carbaminohemoglobin, and He ions from carbonic acid dissociation.
So when oxygen leaves hemoglobin in the tissues, it actually helps the blood pick up more Ki-Ros.
Precisely. And conversely, when hemoglobin binds oxygen in the lungs, it becomes a stronger acid, releases He ions, which helps drive the bicarbonate reaction backwards to produce CROs, and has a lower affinity for binding SHIO directly as carbaminohemoglobin. It helps unload Ki-Oro.
So the Bohr effect, Ki-Oro affecting OREO release, and the Haldane effect, OREO affecting CoUR pickup release, work together synergistically.
Beautifully synergistic. They optimize both oxygen delivery and carbon dioxide removal at the right places.
And the net result for key OEO removal.
According to the NCERT, every 100 milliliters, 100 ml of deoxygenated blood delivers about 4 milliliters of CRO to the alveoli as it passes through the lungs.
Okay, we've covered the mechanics and the chemistry. But this whole system doesn't just run on autopilot at a fixed rate, does it? Our breathing changes depending on what we're doing.
Absolutely not. We need to be able to adjust our respiratory rhythm constantly to match the metabolic demands of our body tissues. If you start running, your cells need more oxygen and produce more carbon dioxide, so your breathing must increase. This regulation is managed primarily by the neural system.
Where is the main control center located?
The NCERT identifies the primary site as the respiratory rhythm center, located in the medulla oblongata region of the brainstem. This center is responsible for generating the basic rhythm of inspiration and expiration.
So it sets the fundamental pace, like a pacemaker.
Sort of, yes. It generates the cyclical nerve impulses that cause the diaphragm and intercostal muscles to contract and relax. But this basic rhythm can be modified.
Modified by what?
There's another important center located nearby in the pons region of the brainstem called the pneumotaxic center.
Pneumotaxic center, what does it do?
Its signals can influence the respiratory rhythm center. Specifically, the NCERT says its signals can reduce the duration of inspiration.
If you shorten the inspiration time, you automatically increase the breathing rate, right? Breathe faster, shallower breaths.
Exactly. So the pneumotaxic center acts as a modulator, helping to fine-tune the respiratory rate and pattern. There's also thought to be an apneustic center in the pons that might prolong inspiration, but the pneumotaxic effect on rate is clearly stated.
Okay, so we have a rhythm generator in the medulla and a rate modulator in the pons. But how do these centers know when to adjust the breathing rate? What information are they responding to?
This is where chemical feedback becomes crucial. Located adjacent to the rhythm center in the medulla is a specialized chemosensitive area.
Chemosensitive, sensitive to chemicals. Which chemicals?
This area is highly sensitive to changes in the levels of carbon dioxide, CO euros, and hydrogen ions in the cerebrospinal fluid bathing the brain.
Not oxygen.
Primarily CO and HO. If CO levels in the blood rise, CO crosses the blood-brain barrier, gets converted to carbonic acid in the cerebrospinal fluid, and releases AHO ions. It's these HO ions reflecting the CO level that strongly stimulate this chemosensitive area.
So if you start exercising, produce more CO, the He level near this area goes up.
And this chemosensitive area gets activated. It then sends signals to the respiratory rhythm center, telling it to increase the rate and depth of breathing.
Why? What's the goal?
The goal is to blow off the excess COers more quickly, which in turn reduces the HAO concentration and brings the pH back towards normal. It's a classic negative feedback loop to maintain blood gas and pH homeostasis.
Is this the only place the body senses these chemical changes?
No, there are also peripheral receptors. The NCERT mentions receptors associated with the aortic arch, the big artery leaving the heart, and the carotid artery, the major artery supplying the brain.
The aortic bodies and carotid bodies.
Yes. These peripheral chemoreceptors also detect changes in blood COO and HO concentrations. They can also sense significant drops in blood ORS levels, which the central chemosensitive area doesn't really do.
Ah, so the peripheral ones do sense oxygen.
They can, but their sensitivity to CO, and HO is generally considered more important for routine regulation. When these peripheral receptors detect changes, especially increases in CO or HO or a large drop in OO, they send nerve signals back to the respiratory rhythm center in the medulla to trigger corrective actions, usually increasing ventilation.
This brings us to a really interesting point the NCERT makes. Despite oxygen being the reason we breathe, its role in regulating breathing seems secondary.
It's quite surprising, isn't it? The NCERT explicitly states that the role of oxygen in the regulation of respiratory rhythm is considered quite insignificant under normal circumstances compared to the powerful influence of CO and HO.
Why would that be? Why isn't the body constantly monitoring the fuel level as its top priority?
Well, think about it. Under normal conditions, hemoglobin is already nearly saturated with oxygen leaving the lungs. Small fluctuations in breathing don't change oxygen uptake dramatically unless you're already compromised or at high altitude. However, co-urose production is directly linked to metabolic rate, and even small accumulations can rapidly affect blood pH because of the He ions generated.
So maintaining pH balance by getting rid of KEO is the more immediate, critical regulatory challenge.
Exactly. The body prioritizes keeping the internal chemical environment stable. The drive to breathe is primarily driven by the need to eliminate the acidic waste product, Q euros, rather than the need to acquire O, although obviously acquiring O is the ultimate goal. The co-air host system provides a much more sensitive and responsive control mechanism for matching ventilation to metabolic activity.
So we have this incredibly complex, finely regulated system, but like any complex system, things can go wrong. The NCERT touches on a few common respiratory disorders.
Yes, it highlights some key examples where parts of this system fail or become diseased. First, there's asthma.
Asthma is quite common. What's the underlying issue according to the NCERT?
It's defined as difficulty in breathing that often causes wheezing. This is due to inflammation of the bronchi and bronchioles, the conducting airways. This inflammation makes the smooth muscle in the airway walls hypersensitive, causing them to constrict easily in response to triggers.
So the airway is narrow, making it hard to move air, especially out.
Exactly. That constricted airflow causes the characteristic wheezing sound and shortness of breath.
Okay. Another disorder mentioned is emphysema.
Emphysema is a chronic, often progressive disorder. The core problem here, as the NCERT states, is that the alveolar walls are damaged.
The exchange surfaces themselves get destroyed.
Yes. The thin elastic walls break down, leading to larger irregular air spaces instead of the millions of tiny efficient alveoli. This results in a significant decrease in the total respiratory surface area available for gas exchange.
That sounds serious. What causes this damage?
The NCERT points to one major culprit very clearly: cigarette smoking. The chemicals in cigarette smoke are incredibly destructive to the delicate lung tissue, leading to the breakdown of elastic fibers and alveolar walls over
A
time.
largely preventable cause, then.
Very much so.
Finally, the NCERT discusses occupational respiratory disorders. These relate to workplace exposures.
Yes. People working in certain industries, particularly those involving grinding or stone breaking, are exposed to high levels of dust over long periods.
And the lungs' defense mechanisms can't cope.
Exactly. The sheer amount of inhaled dust overwhelms the normal clearing mechanisms like mucus and cilia. This chronic irritation and particle deposition leads to inflammation.
And that inflammation causes long-term damage.
Yes. It can lead to fibrosis, which the NCERT describes as the proliferation of fibrous connective tissue in the lungs. Essentially, the lung tissue becomes scarred and stiff.
Scarred and stiff, that would make breathing difficult and reduce gas exchange efficiency, right?
Absolutely. It leads to serious, often irreversible lung damage, reducing lung compliance and overall capacity.
Is there a key takeaway for prevention here?
The NCERT offers a clear recommendation. Workers in these high-risk industries must wear protective masks to minimize dust inhalation and prevent the development of these debilitating occupational lung diseases. #, OUTRO, synthesis and takeaway.
Well, that was quite a journey through the respiratory system, following the NCERT's path. We went from basic anatomy, like those incomplete cartilaginous rings,
Then the crucial pleural fluid.
through the physics of breathing, the mechanics of the diaphragm, and intercostals creating pressure gradients.
Then into the detailed physics of partial pressures, the surprisingly vital role of co-uro solubility.
And on to the intricate chemistry, hemoglobin's reversible binding, the elegant oxygen dissociation curve, the Bohr effect.
And the complex multi-step process of co-uro transport involving carbaminohemoglobin, carbonic anhydrase, bicarbonate ions, the chloride shift, and the Haldane effect. It's quite the chemical dance.
And finally, the neural regulation, the rhythm center in the medulla, the neotaxic fine-tuning from the pons, and that critical feedback loop driven primarily by CODIO and HIF levels detected by chemosensors.
For me, that regulation piece really underscores the systems biology perspective we started with. The sheer precision, tiny changes in waste products triggering immediate adjustments in ventilation shows how elegantly the body maintains its internal balance, its homeostasis. It's not just about individual parts, it's about the controlled interaction.
It really is remarkable. So to leave you, our listener, with something to ponder, let's connect back to that regulation and the real world. The NCERT kind of prompts this by mentioning disorders, but think about environmental challenges.
Like altitude.
Exactly. The NCERT brings up hypoxia-oxygen deficiency. So here's a provocative thought. What happens when you go up a high hill or a mountain? The atmospheric pressure drops, and therefore the partial pressure of oxygen PO in the air you inhale drops significantly.
Right, the driving pressure gradient for oxygen uptake is reduced.
But we just established that the body's primary breathing regulation is driven by KO-ERO, and the role of OERO is normally insignificant. So, when that atmospheric PO plummets at altitude, what happens? Does the body initially not respond strongly because KO-EROs might be normal? How does the system eventually adapt? What mechanisms kick in when the fuel supply itself becomes the critical limiting factor, overriding the usual waste-driven control?
That's a great question. How does acclimatization work at the level of gas transport and neural control when faced with chronic low PRO? Something for you to explore further.
Indeed. Food for thought until our next deep dive.
Thank you for joining with us today for this deep dive into the NCERT. See you next time.
See you next time.